Good afternoon, good evening, everyone, from whichever time zone you're joining us. And on this edition of the Sharing Progress in Cancer Care, behind every drug, there is a story, bringing the human experience back into drug development. We are truly deeply honored to have with us the man, the doctor, the scientist, the drug developer, who has, and I'm not overstating this, literally changed the face of oncology as we know it. Dr. Brian Druker, welcome.
Thank you for having me, Alex.
Absolutely, it is our pleasure. It is my pleasure. So, Dr. Druker really needs no introduction. His legacy is something that precedes him, and I am still going to read his bio, so buckle up. Brian Druker, MD, is the Chief Executive Director of the Oregon Health and Science University Knight Cancer Institute, Associate Dean for Oncology at the School of Medicine at the Institute, and the GELDWEN Chair of Leukemia Research. His research is focused on translating the knowledge of the molecular pathogenesis of cancer into specific therapies and investigating the optimal use of these molecularly targeted agents. He performed preclinical studies that led to the development of imatinib, Gleevec, for chronic myeloid leukemia, and then spearheaded the highly successful clinical trials of imatinib, which led to the FDA approval of the drug in record time. This work changed the life expectancy of patients with CML from the average of 3 to 5 years to a 95% 5-year survival, and has resulted in a paradigm shift in cancer treatment from nonspecific chemotherapy to highly targeted therapeutic agents. He is a member of the National Academy of Medicine, the National Academy of Sciences, and among numerous awards is the recipient of the 2009 Lasker-DeBakey Clinical Medical Research Award and the 2012 Japan Prize in Healthcare and Medical Technology and the 2019 Schoberg Prize. Dr. Druker, anything to add to this?
I did. Thanks for reading through that. It's been just a remarkable ride to see what's happened in oncology from when we started out with non-specific chemotherapies, and now we have all these amazing targeted therapies, immunotherapies, and most of all, what we're doing is we're giving patients hope, and that's all what oncology is all about.
And I love that opening so much. We're giving patients hope. That is what oncology is all about. And you have only a little bit to do with that. So, Brian, I wanted to take us back in time a little bit, if that's okay with you.
Sure.
And as you feel appropriate for our audience, would you tell us a little bit about your formative years, perhaps when Brian was a little boy? Those moments, some of the moments that are particularly relevant for you growing into the man that changed the face of oncology.
Yes. Well, thank you. And some of my first memories as a child were I was incredibly curious. And I remember being like 3 years old, and I'd follow my mom around, and every question that came out of my mouth was why? Why this? Why that? What's going on there? And I just had incredible curiosity. And as a young boy, I had a reputation in the family for, I always wanted to figure out how things work, a clock, a watch, a radio. And I'd take things apart, but I could never put them back together, which is why I never wanted to could never be a surgeon. But I just wanted to, well, how does this thing work? What makes that clock tick? And growing up as the youngest of 4 kids, my father was a chemist. He had a chemistry set down in the basement he would never let us use because he knew we'd blow up the house. But I grew up in a family that was passionate about education, passionate about moving forward in our career, because my father was a first-generation immigrant. My mother was second generation, and they firmly believed that education was the path forward. And they desperately wanted a doctor in the family. Now, my oldest brother grew up in the 60s, went to college, and as he said in college, he had too much fun and he flunked out.
Mhm.
My next brother wasn't particularly interested in science. My older sister wasn't particularly gifted in science, and I was the last hope. But for me, science and math came naturally. And I have to admit, during high school, I was kind of a quiet, reserved, nice guy, kind of a geek. But I
Mhm.
was always the person people wanted to sit next to help them on their homework, on their tests, on anything, because math and science just came naturally to me. So that was me growing up. I love being outdoors. I love sports. I had no talent, uh, but ultimately I followed my talents, which are math and science.
In science, math and science. I'm hearing a very curious boy in there.
Very,
And
very curious.
one that seemed to have carried that torch um, that got passed on from your parents around how important education would be, how important it would be to be a doctor at the end of the day. Um, in addition to the gifts that I'm sure you received with that torch being passed on to you, was there a burden of expectation that you carried with you as well?
Oh, absolutely. My parents, I'd bring home a report card with 5 A's and 1B, and the question is, what happened? What happened there? And there was always very, very high expectations in my family to achieve.
Yeah.
And again, and of course, they were looking out for me. They wanted me to be successful. But they pushed, and they pushed pretty hard because they wanted to instill a work ethic in me.
Mhm.
But for me, being persistent came kind of naturally. Being curious has driven me, and
Mhm.
the thread that goes throughout my life is I always wanted to figure out how things worked. And it was always, if you figured out how things work, you might be able to fix it. So that, again, that
Exactly.
was always that thread of curiosity, persistence, instilled with a work ethic, and all those things came together pretty nicely.
They really did, didn't they? I'm
Yeah,
just wondering that little Brian, as you moved through your school, the early days, and then high school, and then later on college, etc. With that level of expectation, and what happened when there would be 1B on the report card, what did that younger version of yourself perhaps make that mean about him?
I guess I've always viewed the world through that lens, which is, can you make something better? And if you look at something, are you satisfied with where you are, or is there something you can do that will make it better? And I think that's the lesson that has carried through in my life, which is, you can be the best at something, but there's a famous quote from one of my donors who has an incredible story. Her name is Gert Boyle, and
Mhm.
she was the CEO of Columbia Sportswear here in Portland, Oregon. And there's a sign on the wall which says, "That's perfect. Now go make it better." And yeah, to me that just encapsulates kind of that view of, are you happy with what you've got, or can you make it better? Can you make this world a better place? Can you take cancer trials? Is chemotherapy good enough? Or can you do better than that? And how are you going to do better? Well, let's figure out what makes cancer cells tick, and
Mhm.
maybe we can figure out how to attack them specifically.
Yes, yes, exactly. So, would you bridge for us, please, that time period from the report card that may have had just that 1B, and then ultimately you landing your first job in oncology? How did that
Yeah.
happen?
Well, there was a lot of time in between there. There was
Okay.
college. A great
Mm-hmm.
story about college. I grew up
Yeah.
in St. Paul, Minnesota, in the middle of the northern Midwest, and I had all stretched colleges: Harvard, Yale, Stanford, Princeton, Dartmouth, and I got rejected everywhere. And I was a pretty good student. I graduated in the top 5% of my high school class. I had reasonable SAT scores. But At the time, I graduated from high school. I didn't have any college acceptances. My backup was to go to the University of Minnesota. And all you had to do was show up with a better than a C average and you got in. My brother, who had preceded me to California, said, hey, there's this new school down in San Diego, UC San Diego. They've got this great pre-med program. I know that's an interest of you. So I applied, I got in. My choices now were UC San Diego in La Jolla, California, or St. Paul, Minneapolis, Minnesota. Tough choice. I went to California and I loved it. It was a great school. I worked in the one summer in the lab of John Abelson on molecular biology. And what I learned about myself was that was something that I had a natural talent for. Now, this was the day before my first job in the lab. I was the kid from Minnesota. They stuck me in the cold room purifying restriction enzymes because there was no New England bio lab where you could just buy them. We had to purify our own. And for me, looking at the palindromes in the genome to identify restriction enzyme sites, I was natural at that. I was good at cloning and figuring out where to make the cuts and pastes. And I took a molecular biology class, and I was tutoring half of the students in the class. It was just, and I was getting 100% on the test without doing much studying. So I discovered a natural talent that I had that I didn't really know, and so I pursued that on to medical school. And for my first year of medical school, I took a class on the history of chemotherapy. And there's a really critical thread here because the class that I took was on the cure of childhood leukemia. And every article started with, from the 1950s, started with childhood leukemia is the most common childhood cancer. Average life expectancy is 6 weeks. And all we can do is our best to relieve the intense bone pain that these children experience. That was the way every one of those articles started. And then in the 1950s, Sidney Farber introduced methotrexate, and it doubled survival to 12 weeks. And then by the 60s and 70s, combination chemotherapy came along, and you cured this leukemia. And I was fascinated by that. You could cure leukemia, but I also looked at it through that lens of, can you do better? You're inflicting 2 years of toxic chemicals on these poor kids, and there's got to be a better way. So in the final essay that I wrote for that class, I wrote, and I was, again, a first-year medical student, I wrote, only by understanding what drives the growth of cancer cells can we devise better cancer treatments. And that set me on my path. And
Talk
that's
about
what,
being,
yeah.
yeah. Talk about being ahead of your time.
I saved the paper from that class only because the instructor wrote on it, Brian, it's been a pleasure having you in my class. I hope that you follow this passion. It's like, oh my goodness, somebody said something nice about me. It was, as I treasured that more, not for what I wrote, but for the fact that somebody said something really good about my participation in the class.
Right, right. And it's that really innate need in every one of us to feel seen and to be heard. And
Yes,
get the recognition of your gifts and everything that your intention represented.
yes.
Right.
So after that, I did my internal medicine training at Barnes Hospital, Washington University in St. Louis, and then I moved on to Dana-Farber Cancer Institute, where I worked in the same building as a researcher that Sidney Farber had done his seminal work. And I found my way into the lab of Tom Roberts and was working on tyrosine kinases.
Ah.
And it was just, I picked a lab. I knew that I wanted to work on oncogenes.
Uh-huh.
I'd heard great things about Tom Roberts, and he took this medical oncology fellow who had very little laboratory background and took a chance on me. And I worked really hard. I had to learn a whole new language. I remember reading this Scientific American article written by Harold Varmus and Mike Bishop. And they talked about acutely transforming retroviruses. And I remember reading that article and thinking, acutely, I get that. Transforming, I kind of understand that. What's a retrovirus and what do those words mean? It was a completely foreign language. And so, but I made my way, I worked hard, I made my way along and developed an antibody that was actually a phosphotyrosine antibody that lots of people used. And it exposed me to immunology and platelet biology and neutrophil signaling and all these signaling pathways. And so I was gaining a lot of expertise in the field of kinases and signaling. And in 1988, a group at what was then Ciba-Geigy, ultimately Novartis, headed by Nick Leiden, came to visit. And at the time I was working on a polyoma virus model. It was a rat model of cancer, polyoma middle T virus, which now we know is involved in Merkel cell carcinoma. But at the time, it was just a protein that brought together Src family kinases, PI3 kinase, and I was working on its transforming capabilities. Nick Leiden came and visited, and we were talking about what targets they might work on. And I said to him, Nick, the first target that's going to fall to a kinase inhibitor is going to be chronic myeloid leukemia because it's driven by the BCR-ABL tyrosine kinase. That was 1988. In 1990, I went out on a long bike ride, and I was thinking, what am I going to do with my future? I'm working on kinases, I'm making some progress, and I was so distracted, my wheel got caught in a railroad track, and I took a massive fall. So I got up and I said to myself, this is crazy. I'm out on this long bike ride. I need to focus. I need to change my work to chronic myeloid leukemia and BCR-ABL because I have a background in kinases. I've said that BCR-ABL is going to follow the kinase inhibitors. That's what I should be working on. And so I forged a collaboration with Jim Griffin, and I started working on BCR-ABL and CML.
That is fascinating, and I would really love to pull a thread from a point earlier in our conversation to the bike ride and the fall.
Yes.
So as I was hearing you speak about choices for college and all of those, let's call them rejections on a human level, and how you ultimately landed in San Diego and not in Minnesota, and where then your career and your natural innate talents took you, how you understood so intuitively, so naturally, so intrinsically and organically things that perhaps were hard for other people, but for you, you were breathing it. It feels like you were living it. And then all of the experience in the steep learning curve around oncology, that whole arc from chemotherapy to TKIs, immunology, stories of leukemia, and that it was just not good enough, to the bike ride, the fall, and I should be working on BCR-ABL and CML. Now, please correct me if I'm wrong, but I'm noticing a path with perhaps a presence of, let's call it something bigger than us. Perhaps you were, as if you were always meant to be where you landed, and life had a way of putting you exactly at the right place at the right time. What is your reflection as you hear me saying this?
Yeah, I know. It's so interesting, Alex, to hear you say that because I always have looked back at my life as kind of a river with lots of twists and turns and bends, but I landed in the right spot. And I've always looked at it that way. I kind of went in with no clear guidance and direction, but figured out where I where I was at the right time and the right place. And it all came together, as you said. And I've just always been, it's always been fascinated how sort of it's always kind of like fate, whether you believe in it or not. Uh,
Mhm.
but I landed in the right spot at the right time. But more importantly, you know, one of the stories I tell often in my talks is about the discovery of Velcro.
Mhm.
And it's a story of, it's not always, people say, oh, you've got to think outside the box.
Mhm.
And for me, it's sometimes you got to just look at what's right there in front of you and see it and move it forward. And the story of Velcro is this guy, George de Mestral, went out for a hike in the woods and somewhere in the Alps, was wearing a velvet vest and came back with his vest covered with burrs.
Mhm.
And how many people throughout history have gone on hikes, gotten their clothes covered with burrs, picked the things off their clothes, got stuck on their finger, and swore at the things?
Yeah.
But he was an engineer, and he said, why do these things stick so tightly to my velvet vest? So he found a microscope, looked under the microscope, and his vest had loops and the burrs had hooks.
Yeah.
And he said, I bet I could make a fastener out of that. So he asked the important question, why?
I love that. I love that story. There are so many parallels throughout walks of life if we're just willing to see them.
Yes, that's right.
Yeah. And I wonder throughout this journey that you just brought us current to the light bulb moment, and that probably the way I'm actually hearing you say this and the way it's landing for me is that deep knowing inside I should be working on BCR able and CML. It's like that knowing, the unshakable, unmovable. I just know this in my bones.
Yeah.
Um, Up until that point and going forwards, what were your lessons around, or maybe what was your relationship to the word yes and the word no? Because I
Yeah.
imagine you've heard both quite a bit.
Yes. Well, there's two important stories here.
Okay.
And clearly, this is the one that's going to put it over the top in terms of what motivated me.
Mm-hmm.
And I'll tell a story about, my wife is a writer, and she loves to write sort of provocative talks that I give.
Mm-hmm.
And so one of the talks she had, I was having to give a commencement speech for medical school graduation. And she instructed me, walk up to the podium and start your talk. Do not thank the audience. Do not thank the speaker. Just walk up and say these three sentences.
Mm-hmm.
You don't have a future. There was dead silence. You're not going to succeed. More dead silence. Last one. I hope you never pursue a career in science. And at that point, there was kind of muffled laughter.
Uh-huh.
My next sentence was, those are some of the words of encouragement I got during my career. And the point was, do you believe those words, or do you use those words to, to move forward? And do you use them to shape what you are? And so the most important story that shaped me was I was looking for a promotion at Dana-Farber. A
Mm-hmm.
number of people younger than me got promoted to assistant professor. I met with the head of Dana-Farber, and he looked through my resume, and he was one of the people who basically said, I don't think you have a future here. And at that point, I had a choice. Do I believe in myself, the work I'm doing, trying to understand BCR-ABL signaling, trying to identify an inhibitor of that kinase, trying to move that into clinic? And what I recognized about myself is that I'd always given things about 90% effort, because I always had this in the back of my mind, well, if I only give it 90%, if it doesn't work out, it's my fault, and so I have myself to blame.
Mm-hmm.
But now I decided that I got focus. I went through a period of a little bit of depression because, those are tough words to hear. But I then decided I'm going to find a place where I can go and work on my dream. And I'm going to find a company that has an Abl kinase inhibitor, and I'm going to get it into clinic. And now I had drive, passion, focus, and a purpose, and I was going to make this happen. It wasn't because I wanted to prove that person wrong. It was because I understood what I needed to do to get something done. So I made one call to Nick Lydon, who's at Ciba-Geigy. So I landed at Oregon Health and Science University. I had a wonderful mentor, Grover Bagby, who was developing a cancer program. And he said, Brian, I believe in your work. I want you to move here, and we'll give you a startup package to get this work done. I arrived in Oregon. I called Nick, and I asked Nick, hey, Nick, do you have any Abl inhibitors? He said, well, as a matter of fact, we have one. I've been trying to find somebody to work with it, and I'll send it to you. Now, a lot of people said, oh, I got lucky. But what they don't recognize is that I had a script with Nick. And if Nick said, yes, I have some, I was going to say, can I get them? If he said no, it was going to be, do you know anybody who has one? It wasn't going to be a one call and done. But I did get a bit lucky that I only had to make one phone call. Nick sent me a half a dozen vials with the only information was their molecular weight and no profile, because he wanted me to do the experiments blindly. So with no knowledge of how to do these experiments, I dissolved these compounds, and there was one that stood out at killing my leukemia cells without harming normal cells. That one turned into be imatinib or Gleevec. And 6 weeks after I arrived in Oregon, I had that data. Now, it took me 5 years to get that drug into clinical trials, and there's a whole story there about convincing a big drug company to take a drug that will never make back their investment into clinical trials, but ultimately we did.
And we'll get right to that. I would just love to reflect on this last story you told us. And there is a quote that came to mind. One of my favorite authors, Matt Kahn, and the message you heard from your colleague at Dana-Farber and how that redirected you to Oregon. And the quote is, the human perception of rejection is the soul's awareness of redirection. And
Yes.
that to me sort of encapsulates that pivot from Dana-Farber to Oregon.
Yes.
And then how I heard your story in Oregon was that, and this goes for just life in general, and certainly drug development, it is a part of life. This is something that moves through people, and that's really what I want our viewers and our listeners to feel. This drug moved through you.
Yeah.
And often there's a certain level of consciousness that is required, and an awareness and a lot of pieces that need to come together that clicked within you, but that also need to be recognized at the matched level of consciousness, if you would, by that someone who will say yes, who will believe.
Yeah.
And it was that colleague of yours at Oregon who said, Brian, I believe in you.
Yeah.
And here's a startup package. And we
Yes.
need that support.
Yes, we do.
Support.
You need people at times in your career that will just put their arm around your shoulder and say they believe in you. And
In
that's
your
so
career
critical.
and in life.
Yes. Yeah. Yes.
Yeah. So tell us, how did you ultimately get to be Gleevec in clinic?
Yeah. Well, first of all, my clinic was now becoming more and more directed toward patients with CML. And I remember it was 1996, and I had this young man in my clinic, and he was a computer and tech guy. And he had this vision for developing a new computer software, a new search engine that was better than was out there. That was the same year that the Google guys were beginning to think about a better search engine. So he was right at the right place at the right time. His leukemia, CML, had just transformed to blast crisis, and he died. And this could have been the founder of Google sitting right there in my clinic. And those patients would prompt me to make a call to my colleagues at Novartis and say, why aren't we taking this drug into clinical trials? And the answer was always, well, we have this hurdle or that hurdle, or there was always an excuse. And what I learned later is the excuse really was, if you did a market survey, as you well know, it takes $1 billion or more to develop a drug, and your rate of success is maybe 1 in 10. And so you set the bar for your return on that investment at about 500 million a year. Otherwise, you'll never make back your investment or the 9 other drugs that failed. When they did their market analysis of, we'll take the current drug, which is interferon, we'll replace it at about 20,000 a year. We'll have 30% market penetration in the first year. That comes out to 100 million a year for the population size of CML. So would you invest $1 billion in a drug that has a 1 in 10 chance of success, and a kinase inhibitor has never been developed anyway, and it may have some toxicity, probably won't work, and it's never going to make back your investment? Would you invest in that drug? You'd be crazy. So ultimately what I learned, and I wish I'd become a parent sooner, is that when I ask my kids, what do you want for dinner? They'll say, I don't know, what do we got? If I ask them, do you want a pizza or a hamburger? 9 times out of 10, they'll make a choice. 1 time out of 10, say, I don't want either of them, but at least, 9 times out of 10, I'll get an answer. And my point there is, if you give people a choice instead of an open-ended question, they can give you an answer. So Nick Lydon, who had actually left to start his own biotech company, gave me some advice. He said, what if we approached Novartis and offered to license this drug out? So now they have a choice: take it to phase 1 trial or license it out. We gave them that choice, and they decided to go to a limited scale phase 1 trial. And their point was, it'll probably fail. We don't have to invest that much money. If it works, as the usual 20% response rate, we can license it off and offload it to a biotech company at that point.
So you mitigated their risk.
Yeah, you de-risk it, you give them a choice, you give them a path forward, and they took it. And so in June of 1998, we started phase 1 clinical trials.
June 1998. Brian, do you remember the day when the first patient got dosed with imatinib, and how did you feel?
Oh my goodness! There was a camera crew from one of the local TV stations came in. The patient was a railroad engineer from out on the coast of Oregon, and he was a ham. And I was checking on him every hour on the hour to make sure he was okay. And he'd come in and say, I'm still here, doc. Still here, doc. I'm okay. And he was just a wonderful, willing participant. And it was just a remarkable, exciting, but frightening day. I'd worked on some of the phase 1 trials of some chemotherapy drugs like Taxol, and we made patients incredibly sick when we hit maximally tolerated doses. So I was excited but nervous about this.
I'm excited. That patient really, the patients that say yes to us in clinical trials, it never goes unnoticed, the level of trust they put in us, in both physicians and the people who stand behind the drugs, and that integrity and that intention that fuels drugs such as imatinib, I believe really is felt by the patients when it is truly, truly genuine, as it was in your case and in the case of imatinib.
Yes.
And it's such a moving story.
And remarkably, this first patient, when we had published our laboratory studies, actually in 1996,
Uh-huh.
He he had contacted me because he read a newspaper article and said, "I have CML. I want to be your first patient." And he was 2 years
And
later.
he was.
Yeah.
And as the trial went on, can you share with us the moment when you just knew, perhaps even silently, but knew, just like you knew on that bike trail, when
Yeah.
you knew this was going to work?
Yeah. Well, I'm going to give you two stories if you don't mind, and
I don't mind at
I'll
all.
try to be brief.
No, no, please
So,
go ahead.
so very early on, as we did our dose escalation,
Mhm.
we had taken patients off their interferon, and we had two types of patients. One whose blood counts would go very slowly, 10, 20, 30, 40 over the course of the next month or two. And some whose blood counts would explode, 20, 100, 200. For the very early dose levels, I only put the patients on the first category because I knew I wanted to get them through that first month in case the drug didn't work so we could go on to the
Mhm.
next dose level.
Mhm.
But by 200 milligrams, we were seeing people's blood counts stabilize, just a hint of activity. So 250 milligrams, I decided to be a little bit more aggressive, throw a little bit of caution to the wind. And I put one of those patients on whose white count had gone from 25 to 125,000 in a week off therapy.
Uh-huh.
A week on treatment, her white count was stable at 120,000. 2 weeks later, it was 80,000. 3 weeks, it was normal. And at that point, it was, wow, this is somebody who had failed every other treatment. And this drug restored her blood counts to normal in 3 weeks. By the next dose level, everybody's blood counts was returning to normal. And now I was walking on eggshells.
Yeah.
What if it doesn't hold? What if it only keeps their blood counts normal for a month or 2 months?
Mhm.
Nobody's going to care.
Yeah. Yeah.
It's going to be a fancy hydroxyurea. It's just not going to go anywhere. But I walked into clinic that one day and had 3 patients in a row, and every one of them had the same story. They were feeling well, their blood counts were normal, and their hope for the future had been restored. And they told me they were in tears, I was in tears, and I knew that if they believed in it, I had to believe in it too. And years later, when I was being interviewed by my future wife, who asked me, "What did you learn from your patients?" What I learned from them is the greatest gift is hope. And that was the gift that I had given them that day in clinic, and that's what they shared with me.
And you received it back.
Well, but what I've received back 25 years later, some of those patients are still with me. I've gotten to see weddings, graduations, grandchildren, all the life events and the things that they've gotten to enjoy that they share with me. And to me, they've given that a gift back to me, which is I've gotten to see what effect I've had on people's lives and the benefits they've gotten from the work that we've done. And that's a gift that is just, I feel so blessed to walk into clinic and hear those stories, or to get an email out of the blue from a patient who's read about the development of imatinib and has thanked me. And they always know when I've had a bad day because I'll end up with an email in my inbox, and it reminds me of what we can accomplish by continuing to work hard, be persistent, ask the important questions, and work for our patients.
And work for our patients and believe.
Yeah.
Your belief truly through this story has been unshakable.
Yes, it has. But it has to be.
Unshakable. It has to be. It really, really has to be. Imatinib has grown up quite a bit
Yes, it
from
has.
that toddler back in the day. Tell us about imatinib today.
Well, it's so interesting that you talk about it in terms of growing and developing children. I remember When it got FDA approved, I felt like I was sending a child off to college. It was going
Mhm.
out into the world. Anybody could prescribe it. And that was remarkable.
Yeah.
And now, 25 years later,
Mhm.
I think the two things it's taught us are, number one, you know, it was the birth really of targeted cancer therapies. But the other thing it did within industry is that if you go back to the market, within the first year on the market, it brought in $1 billion of revenue for Novartis. And the reason it did that is not because they priced it basically the same as interferon, so it replaced the current medication, but it had a 90% market penetration in the first year. You could, if a marketer had predicted that, they would have been fired,
Yeah.
but it did. People wanted this medication, and they came off their current drugs, and everybody went on imatinib. But what it meant to the drug industry is you could develop drugs for rare indications, and there was a path forward. And it actually energized the drug industry to think about drugs for rare diseases in a different light. If they worked and they worked well, they could actually make their investment back. And now you've seen lots of drugs being developed for rare indications, eh, maybe in part because of the success of imatinib.
Yes. Hearing you say that, it really punctuates the fact that your journey and story with imatinib really has pierced through so many glass ceilings
Yeah.
and has opened up so many avenues for oncology, but not only for oncology, for
Yeah.
other medical disciplines as well. So this is pioneering work on so many levels. And I will really encourage the audience to go back and re-listen to this interview several times to really capture the gems that you have sprinkled throughout. And now when you think about imatinib, tell me the first two words that come forward.
Ah, I don't know. It's so hard. It's been such a remarkable journey, and I'm just so grateful. For me, I've just been grateful to have played a part in this. And I said that in my first interview when the clinical studies were announced. I'm just grateful that I was at the right place at the right time and put these pieces together. But I also recognize that it's a linear progression. And I'm sorry I'm not coming up with two words because I can't think of two words. But I put a piece of the puzzle in, and it was a big puzzle. It was the last piece, but I couldn't have done it without the work of decades of progress. And to me, we all play our part. We all put a piece of a puzzle in, and sometimes that puzzle gets solved. And that's the beauty of science, is we follow the science, we make a contribution, and sometimes we don't know where it's going to go. Sometimes we solve a puzzle.
I hear two words in that still, and I hear the words gratitude and trust.
Yeah. Thank you.
Yeah. What would you say was the biggest low and what was the biggest high on this journey?
Yeah. The biggest low really was not getting a promotion.
Mhm.
Uh, and that took me a long time to recover from that. But it also, as I've said, it also, when you look back on it, it was a gift. And it was a gift that gave me that extra gear that I needed to push me over the top. So, you know, if people said, what would you change? I wouldn't change a thing. I needed a kick in the pants at that point, and I got it. And
Mhm.
so I kind of look at it back as a gift. Um, but the greatest high was that day in clinic when people shared their gift, the gift of hope and their trust that they had put in me. Patients had put their, as you said, had put their trust in me that I was going to do something that was going to help them. And to live up to that trust that had been placed in me was just a remarkable, uh, accomplishment and something I'm incredibly grateful for.
Indeed, indeed. And you continue to live with such integrity and to bring
Thank you.
that to oncology and to life into your patients every single day. I'm curious, as we're wrapping up, were there any compromises that you had to make along the way, be it in your professional or your personal life, that you're
Yeah,
willing to talk about?
yes. Well, I'm always fully open and transparent, and I'm on my second marriage. My first marriage failed because I worked too hard. I worked long hours. I was at Dana-Farber. I was trying to get ahead in
Mhm.
my career, and I spent all my waking hours working and not paying attention to my wife. And the marriage didn't work. And I regret that, and I recognize that about myself. And but I've learned.
Yeah.
I've learned that when people ask me about work-life balance, you know, I reflect on that. And I've said, you can maybe do two things well at once. You can work and you can have a family, but you need to focus intensely on both of those.
Mhm.
At the time, even at that point, so when I was working on the development of imatinib, my future wife who interviewed me asked me, well, can we talk to people that know you well? And I was, well, right now I'm working really hard. I was actually doing some competitive amateur cycling. And so I'd work out, I'd eat, and I'd sleep. And she looked at me and said, you're pathetic. So after her story was published, I got the guts to ask her out on a date. We got married. She brought a child from her first marriage, a 3-year-old. We now have 3 wonderful children. I didn't miss one of their soccer games, basketball games, parent-teacher conferences. I was a very present husband and father, but I was still working pretty hard. So I could do 2 things, but not 3.
Right.
But I learned that I had to prioritize my work and my family. And those are the things that I've learned about success and failure. And I think I've done okay with my failures and figured out how to do better. And again, it's
Absolutely.
back to that thread of doing better.
Doing better. Thank you so much for sharing that so openly and vulnerably with us. I really, really appreciate that on a very personal level too. And as parting words, is there anything, Brian, that I haven't asked you that is on your heart that you're wanting to share, perhaps what you're excited about now, and any last advice for people who are perhaps beginning in drug development or are experiencing some challenges along the way?
Yeah. Well, as my wife has written for me,
Mhm.
it's we savor our successes, but we never forget the patience we lose. And that's what keeps me going, is I know what success looks like, but I want that for every one of our patients. And until we get there, we have work to do. But we have incredible science and technology to bring to bear on the problems that we face. And that's why I'm optimistic about the future. We have incredible tools, both with the drugs we have, but also the tools we have to understand cancer and attack it more effectively. So I'm incredibly optimistic about the future.
Amazing. Thank you so much.
Well, thank you for
I
having me.
it has been the greatest honor and my greatest pleasure. And truly, this has been, for me personally, the highlight of one of the highlights of my career, to be in conversation with you today, to learn from you, to receive your story told by you. And I too will be listening to our conversation several times over. I really, really encourage everybody to do so. This is a gem every step of the way. Please, please join us. This has been the crown, the Crown interview with Brian Druker on Behind Every Drug, There is a Story, bringing the human experience back into drug development. And boy, Brian, did you do that today? And have
Thank
you
you.
done that in your life? Thank you for your legacy.
Oh, thank you so very much for having me, and all the best to you in your career and drug development journey.
Thank you.
Thank you.